Hello everyone, this is Higher Biology, and we're going to treat this as we would as if we weren't under lockdown, we're in the classroom. So hope you're settled somewhere, you've got something to write with, something to write on. If you've got a textbook, you could have that beside you, or the notes for Unit 1 that I've put on OneDrive. But yeah, this is Start Higher Biology. Unit 1 is mostly genetics, so it's called DNA in the genome. The genome, remember, is all your genetic information, and we're going to begin with the structure of DNA. So this is the first unit, Unit 1.1, Structure of DNA. Before we get on to looking at the higher material, I think we want to try and establish first of all that you're pretty confident that you've got the basics of DNA from Nat 5. So this is the first time I'm going to ask you to press pause and write down some answers. So see if you can do these numbers 1 to 7 and then press play again and I'll go through them with you. So first one, DNA contains, number 1 is genetic information which the cell uses to make. Number 2 is proteins. DNA, it is made of, number 3 is 2 strands twisted around each other to form a shape known as number 4 double helix. Okay, each DNA strand is a chain of millions of 5 as bases, and one of them is adenine, which is A, and it pairs with number 6 thymine, which is the T. Cytosine, it pairs with guanine, which is G. Okay, so that's the kind of basic stuff from National 5 that and again, just to sort of spell out what we're looking at here, here is an organism, a bald man. Here is one of his cells. Not quite scientifically accurate because he's not just going to have 3 chromosomes in there, is he? He should have 46. But the chromosomes is where the DNA is, and if you were to zoom in the chromosome, then you would have your double-stranded DNA, your double helix, which would look like this over here. Going into a little bit more detail again, so now you can see on these 2 strands you've got your bases, the letters, and let's imagine that you were to zoom in on this little segment here, the A and the T. This is what it would actually look like. And you've got your base, and then you've got these 2 other parts, something we didn't look at in National 5, but this comes into it higher. So think about this area here, and we call this a nucleotide. Throughout these, there's some videos I want you to have a look at. So if you've got access to the OneDrive, you can, and you've got that open, you can click on that. Otherwise, just go into YouTube and search for this. What is DNA and how does it work? Go and have a look at that, and then come back. So learning intentions, and really these 3 cover everything you need to know for the first subtopic of higher biology. So number 1, state what a DNA nucleotide is. I mentioned a couple of slides back, this is our new term, and describe its structure. 2, explain how the 2 strands of DNA are joined together and how they're aligned. So what is the relationship to each other? And number 3, describe how DNA is organised in prokaryotes and eukaryotes. Prokaryotes, eukaryotes, maybe words you may remember from National 5, but we'll look at exactly what that means before we look at the DNA organisation. So here is the nucleotide. You've got 3 parts. Okay, so each double helix strand is made up of these. So it's a chain of these, as we'll see in a minute, and it's got 3 parts. So they are a phosphate group, a base, so that's your A, T, G or C, and then the main part right in the middle is the deoxyribose sugar. So if you think DNA is deoxyribonucleic acid, so you might have wondered where that came from. So this is it here. So the sugar part of the DNA nucleotide is deoxyribose sugar. And you might have been thinking about what these numbers mean. Well, these relate to the carbon atom. So we've got a sugar carbohydrate, so it's made up of contains carbon as one of the elements that make it up, and you've got carbon 1, 2, 3, 4, and then number 5 sort of sticks out at the phosphate group. Okay, so that's for 3 parts of the DNA nucleotide, and certainly by the time you got to the exam stage, you'd be hoping to be able to draw that out from memory. So put them together, and this is what you've got. So you've got a bond here between one nucleotide and the next one in the strand, and you can see, and this is important, the bond is at the 3 carbon, and that little apostrophe there, we say prime, so that's the 3' carbon. So there's a bond between that and the phosphate group of the next nucleotide. Okay, and these are strong chemical bonds. So strong chemical bonds, they exist between the phosphate group and the 3' of the deoxyribose sugar of the next nucleotide, and this is what we call the sugar phosphate backbone. There's the sugar, there's the phosphate. This is a strong backbone in that strand of the double helix. So that's what one strand would look like, your chain of nucleotides. And just to show you here, there's your DNA and the nucleotides in some detail. And what you'll probably notice is when we put the other strand on, you can see that the sugar is upside down. And on this side, we've got the phosphate at the top, and in this one, we've got the phosphate at the bottom. Again, you can see it here. So because they're arranged in this way, we talk about each DNA strand having 2 ends. So when you've got the phosphate at the top, that is the 5' end, and when you've got the deoxyribose sugar at the top, that is the 3' end. So you can see this strand is running from 5' down to 3', whereas the opposite strand, it's upside down as we said before. So you've got the 3' at the top and it goes down to the 5'. And because of this, we talk about the 2 strands running as anti-parallel, so they're running in opposite directions. And you might have noticed from the diagram, we talked before about the sugar phosphate backbone, which is strong here. We've also got hydrogen bonding, and that is, remember this blue bit here in the middle is the bases. So you've got hydrogen bonding between the bases, that is relatively weak compared to the sugar phosphate backbone. Now, if we were in the class, I would be giving you some materials to try and create a double helix, but we are not, unfortunately. So what I'm going to ask you to do instead is do a drawing of it. So first thing to do is draw out your nucleotide, so the 3 parts. Make sure you number your carbons, and the base, we're going to use guanine and adenine. So start off that, draw your first nucleotide, and then draw 3 more below it. So you'll need probably a big bit of paper, and then draw the other side as well. Kind of tip for you, when you go into number 3, what I would do is just once you've drawn the four nucleotides on one strand, just turn your sheet upside down and do the same for the other one. Okay, so pause, go away and do that. It'll maybe take you 5-10 minutes just to draw it out, and then check back with this video or your notes to see if you've got it correct. So that what we did there is the kind of the DNA structure and the way it looks. We did mention the learning intentions that you've got different types of DNA organisation depending on what the living thing is. Again, I always like to start with a bit of Nat5 revision. So do we know each of these? Top left, which type of cell is it? That is animal. Top right, chloroplast, giveaway maybe there, that is the plant. Down here, we've got pretty much the same as the plant cell, except we're lacking a chloroplast, so that is the fungal cell. And then over here, the one that's just kind of out on its own and is quite different to the others, and that is the bacterial cell. So let's have a look at how the DNA is organised in these different types of cell. And to begin with, another video. So search for that on YouTube. The channel is the Amoeba Sisters, and this will give you a good overview of the differences between prokaryotes and eukaryotes. Okay, here is a big table. This shows you the differences between the organisation of DNA in prokaryotes and eukaryotes. So you can see you've got prokaryotes are the only one that we've got here is bacteria. The other ones are your animals, plants, and yeast. They are all eukaryotes. So this first row, I mean, I think you would be able to get this from Nat5 knowledge, wouldn't you? So bacteria have got no nucleus, whereas animals, plants, and yeast do. Okay. In terms of the location of the DNA, the bacteria have got plasmids, which you remember from Nat5, but also they have got chromosomes in the cytoplasm. So 2 locations of DNA. Animals like ourselves, you know, we spoke at Nat5, we talked about the DNA in the nucleus. You probably know that from S1, I would have thought. However, we do carry DNA in another place as well, and that is in our mitochondria. So we've got chromosomes in the nucleus and in the mitochondria. Plants, well, they've got the nucleus, the mitochondria, and then you can maybe work this out, sort of an organelle within plant cells, which is similar to mitochondria. They also have DNA in their chloroplasts. Yeast, chromosomes in the nucleus and the mitochondria, and also there are some species of yeast which contain plasmids. Okay, so that's the location of the DNA. And then the type of the DNA, you have got 2 options, either circular or linear, and I would hope maybe if you're looking at plasmids for bacteria, then you would be able to work out that bacteria have got circular DNA. Animals in the nucleus, if you think about what a chromosome looks like, that is linear. However, we do, remember, have that second location of DNA, which is the mitochondria, and within the mitochondria, our DNA is circular. For plants, linear in the nucleus, circular in mitochondria and chloroplasts, and then yeast, again, you'll see a bit of a pattern here, linear in the nucleus, and they have got circular DNA in the mitochondria, and remember, some species have plasmids. Okay, so that's your organisation of DNA in prokaryotes and eukaryotes. And just to sort of make it obvious, linear DNA, single, strand in a linear fashion, whereas circular comes back in itself and looks like this. So that's all we mean when we're talking about linear DNA and circular DNA. Now the linear DNA found, where is it found again? Yep, chromosomes. And we've got an image here of a planetary body, and the reason why we've got this is one of the kind of facts about our DNA that likes to be that you quite often see in media and so on, is that one human's DNA stretches well out into our solar system and back 2 times 10 to the power of 13. So if it wasn't all coiled up, then that would be well, in order to fit within our cells, it's got to be coiled up, and also mentioned within the course is how that happens. So here is your DNA strand, and you can think of it as almost like a thread, and it is wrapped around this little bead-like protein, and that's called a histone. Okay, so linear DNA is tightly coiled and packaged with these associated proteins, which are called histones. And that is it. That is all the knowledge really required for the first subtopic: what the nucleotide is, the 3 parts, how the 2 strands of DNA are joined and aligned. For number 1 and 2, I always say to my classes, if you can draw out, as you did before, if you can draw out nucleotides, maybe 4 on each side, and you've got it all labelled correctly, then you've pretty much covered that. And then number 3, the kind of differences between linear, circular DNA, what organelles you find the DNA within prokaryotes and eukaryotes, your bacteria, plants, animals, fungi. Okay, so that's that. In the next part of the video, I'm going to go into the next lesson, which is just kind of revision basically. So you might want to go away and do something else and let a bit of time pass to see if this is in your head, and then come back. Okay, so I'm going to carry on anyway. So the second part of this is just sort of revision to try and recall from memory what you should have learned from this subtopic. Here's the first thing I'd like you to do: put down numbers 1 to 18, and hopefully you can come up with the answers for this. So press pause and do that. So here are the answers. So bacteria 1, no nucleus present. Animals, plants and yeast 2, 3 and 4, they are eukaryotes which do contain a nucleus. The location of the DNA for prokaryotes, you've got chromosomes, plasmids first of all, but you've also got chromosomes and they're in number 5, the cytoplasm. Animals, chromosomes in number 6, the nucleus, and 7, mitochondria. Plants, chromosomes in the nucleus, but also 8, and these can be the way around I suppose, can't they? Mitochondria and 9 chloroplasts. Yeast, like fungi, chromosomes in the nucleus and also in mitochondria, that's number 10. And in number 11, there's a few species of yeast which have plasmids. Final bit of information here, the type of DNA. So prokaryotes, number 12 is circular. 13 animals have linear DNA in the nucleus and they do have circular DNA, however. We do in our mitochondria, that's 14. Plants, linear in the nucleus, circular. 15 again is mitochondria and chloroplasts. Yeast, 16 is linear in the nucleus, and then 17 is going to be circular, and they have circular DNA in their mitochondria, and some species have them 18 in plasmids. Okay, so that's what's the required knowledge for DNA organisation in prokaryotes and eukaryotes. So last part of this, that's the prokaryotes and eukaryote stuff, and the last part of this is blank bit of paper in front of you. Don't look at this video, don't look at your textbook, don't look at any notes, and try and do 1, 2, 3. Okay, if you can do this, then you are sorted. Okay, so number 1, draw a nucleotide with the 3 parts labelled and carbons numbered. Join on 3 more nucleotides to make a strand, and then draw the complementary antiparallel strand with the bond named. Okay, so press pause and then I will show you what it should look like. So here it is. You can see in mine I haven't numbered all the carbons, but that would be 1, 2, 3, 4, and then the 5 sticks out at the phosphate. This is your deoxyribose sugar. The pink thing here is your phosphate. You then get the base. Hopefully you've labelled the hydrogen bonding here. You might have also labelled this as a sugar phosphate backbone. If you did, that's great. And hopefully sort of last thing is you've drawn the other strand sort of upside down, and you've kind of made it clear that at the top of this one, that's 5' end, and you've got 3' end of this one, whereas on the other side, you've got the 5' carbon at the bottom and you've got the 3' at the top. Okay, that is us. Thank you very much.